The choanoflagellate connection. By comparing two existing genomes of choanoflagellates to 19 new genomes that they sequenced and to genomes of various groups of animals, Richter, Fazzuni, Eisen, and King found a treasure trove of new information regarding the deep ancestry of these gene families. Some of their conclusions can be summarized as follows. 1. All animals ranging from sponges and reef-building corals to elephants and humans are descended from a common ancestor that lived over 500 million years ago. 2. Although the first animals consisted of soft-bodied creatures that rarely left any traces in the fossil record, they shared many gene families with modern organisms that allow detailed lineages to be traced. 3. Comparisons of about 1,000 sequenced animal genomes to 21 choanoflagellate genomes reveals clear and striking patterns regarding the origins and conservation or loss of various gene families among diverse animal lineages. 4. Genes that were essential for the development of the innate immune system, which allows animals to detect and eliminate pathogens, arose before the first animals evolved in the last common ancestor of animals with choanoflagellates. 5. Among the 1,944 animal-specific gene families that were identified, genes that are now known to play well-characterized roles in development, including transcription factors and signaling proteins such as TGF-beta, hedgehog, PAX, and SOX, were found. 6. 153 of these animal-specific gene families were found in all but 10% of all animals compared in the study. 7. The origins of animals, choanoflagellates, and the single lineage that includes both were accompanied by the evolution of distinctive gene families. 8. About 372 of the gene families previously thought to be restricted to animals have homologs, ancestrally related genes, in the 19 newly sequenced choanoflagellate genomes. 9. Gene families and pathways critical for biosynthesis of 7 essential amino acids that are present in choanoflagellates were lost in animals that share common ancestors. These amino acids are commonly provided by prokaryotes, such as E. coli in humans, that have symbiotic relationships with animals. These findings, based on phylogenomic analysis, provide abundant molecular evidence for the origin of species and biological complexity based on common ancestry and mechanisms such as natural selection. They not only confirm 19th century hypotheses proposed on the basis of morphological comparisons, but they also represent more rigorous and compelling quantitative evidence. In these data, we find numerous examples for innovations that repurpose existing gene families, as well as the appearance of new families. The loss of gene families, such as those for biosynthesis of amino acids, provided by symbiotic organisms, provide additional evidence that the genes that are conserved, as well as those that are lost, reflect the diverse and changing ecological requirements for capture of energy and nutrients among animals that occupy diverse niches and that have evolved diverse structures that enable them to survive in various ways of life.